It's my extreme pleasure to invite this morning, or this afternoon, I guess it is now, Dr. Elodie Passeport, who's at the University of Toronto, where she's on the Faculty of Applied Sciences and Engineering. Dr. Passeport got her undergraduate in chemical engineering, then got a Master of Science in at the Continental Environmental Hydroscience, in the Department of Continental Environments and Hydrosciences at the University of Paris Tech. And then got her PhD at the same place, and she works on using basically built environments to green infrastructure to help contain and clean up environmental contaminants. So we're really happy to have you here today. So you have the floor, Dr. Passeport.
Thank you. Thank you very much for the introduction. All right, let me move this little window here. Okay. So today I'll give you a little flavor of the type of research and the approach that we use in our group at University of Toronto here. So I'm cross-appointed in the Department of Civil and Mineral Engineering, 51% and 49% from Chemical Engineering and Applied Chemistry. So that means I get my salary from the two places. I teach in both departments, I do service in both departments, and I have the chance to recruit students from both departments as well. So I will talk today about one aspect of my research, which is looking at trace organic contaminants. So organic contaminants present at trace level, so very small amounts in green infrastructure. Okay. So in general, I'm interested in various contaminants that come from anthropogenic activities, and whether it's in urban, agricultural, or industrial areas. As you know, everywhere we use various different chemicals, such as pesticides, pharmaceuticals, personal care products. We use different solvents, we use paints, but we also use different materials, including clothes from which we can generate microfibers. We use a lot of different plastic material that can create microplastics that then enter our environment. And so the use of all of these different chemicals and materials results in their presence in the environment, and that can be in the low nanogram per liter range, for example, for some pharmaceuticals in some rivers, up to the very high milligram per liter range, for example, at contaminated sites. The presence of these contaminants in the environment can have deleterious impact not only on human and ecosystem health, but for the economy as well. They can generate biodiversity losses, be at the origin of various cancer or cardiovascular and respiratory disease, and negatively impact the endocrine and reproductive system. And so I just gave one quick example here, which is a recent example that you might have heard of. In the Northwest US, it was found by groups, researchers there, that salmon, coho salmon specifically, were dying massively. And through very detailed work, a research team there was able to find that this was due to the presence of this one molecule here, which is 6-ppd-quinone, which is not even what I would call a parent molecule. It's a transformation product and oxidation product of this molecule here, 6-ppd, which is used in tire manufacturing to delay the degradation of tires. So not only the parent molecules, all these chemicals and materials that we use, but their transformation products can potentially affect human and ecosystem health. Evaluating the impact of water pollution on the economy, as you might imagine, is very difficult. And so there was a few studies, one, for example, from Pimentel in 2010, who found that the total environmental and social cost of pesticides in the US was about $10 billion per year, which is interesting when you compare to how much was spent in the US on that same year to buy pesticides. A more recent studies on endocrine disrupting chemicals in Europe found that the total health and economic cost was about $200 billion per year. Another indirect cost of the presence of contaminants in the environment is that to get rid of them in wastewater treatment plants or in drinking water production plant, you need to implement what we call advanced treatment technologies, for example, ozone treatment, reverse osmosis, activated carbon, UV light, and these are expensive to implement and increasing costs that ultimately you'll find on your water bill. And so in this context, I think there is a need to develop cost-effective water treatment solution. For example, through green infrastructure of various environmental remediation strategies, which I'll define more in a few slides. So cost-effective water treatment solution in order to intercept these contaminated flows and reduce them before they actually reach our environment. And so when you do this, the first thing that you can do for these various green infrastructure or environmental remediation strategies is to look if there is potential for this kind of technologies to improve water quality. And so you can implement what I call here black box approaches, where you look at the inlet and outlet of some system. For example, construct a wetland, a bioretention cells, which I'll define in a few slides. Or if you look at a groundwater contaminated plume, you can look at the start and end of the plume to see if there is some potential for concentration reduction. But if you want to optimize this system to make them more effective at destroying contaminants and more robust, then you need to employ more kind of gray box approaches where you try to understand the transfer and transformation mechanisms that govern the fate of contaminants in this kind of system. So here I are schematically presented what could be a wetland or a type of green infrastructure where you have some sort of soil or media, some liquid aqueous phase and the air and maybe some plants. So transfer mechanism here means that contaminants that are initially in the water can reach the air, volatilize, or can absorb onto solid surfaces like sediments or suspended particles that might settle down. They might also desorb from these places. Whereas transformation mechanisms or degradative processes usually involve the breaking, sometimes forming of chemical bonds. And this can be due to biotic or abiotic processes. For example, microbial transformation, photolysis, and plants might uptake and transform some contaminants and then either accumulate them or their transformation products and or release them. And so in my work, we contribute or try to contribute at this in our research group to improve water quality and with the goal to protect human and ecosystem health. And my approach is to try and understand these reaction mechanisms so that we can inform on how to best design and operate a variety of environmental remediation strategies. So overall in my research lab, we work on various types of passive water treatment systems for contaminant removal. And this includes, for example, constructed wetlands, with you have an example here. So a pond with some plants, the bioretention cell, which is this kind of urban green infrastructure, which I'll show again later. Some algae pond, but we also work in the context of groundwater remediation. As a matter of fact, I was at a conference all week where it was specifically working at this very highly contaminated site. And on this aspect, my contribution is in the development of stable isotope methods, so CSIA, compound specific isotope analysis. And so we develop new methods for new molecules and we apply them in new contexts. So that's kind of the second theme of my research, which comes in support of overall understanding this green infrastructure and making them better to improve water quality. In terms of the kind of contaminants that I've been working on, my lab is designed to work around organic chemicals. So that's why we've been working a lot on this substituted chlorobenzenes here, but we've worked also with personal care products and other trace contaminants. This is an example of triclosan. You might have heard of it. It's a biocide included into a lot of personal care products. And then when occasion or opportunities arose, I worked on nitrogen, phosphorus. There's a lot of interest on this in Canada due to eutrophication of our lakes. And I've worked on selenium and strontium or radioactive strontium pollution. So I've worked on a variety of areas. And then more recently, in the last 5 years, we've developed all the analytical infrastructure for microplastic monitoring. Okay, but today, in today's talk, I'll focus on benzotriazole. So this is this molecule here, and I'll introduce it in a few slides. And we'll look at its fate in green infrastructure. Specifically, I will talk in the context of two types of green infrastructure, bioretention cells and constructed wetlands. So bioretention cells are a type of urban green infrastructure, which is designed by excavating the natural soil and replacing it with some engineered media. Often this engineered media is basically sand, but sometimes amendments are added in order to absorb contaminants a bit more efficiently. They are vegetated with different types of plants or vegetation, and then often there is mulch at the top here. And the main goal of this bioretention cell is really to intercept and reduce the flows of urban stormwater runoff. You can imagine that in a city where you only have or mainly have roads and buildings and parking lots, when it rains, the water has no place to infiltrate. So it just accumulates and creates this runoff. And so bioretention cell is one of the systems that help control this volume of stormwater runoff and reduce the risk of flooding. And while the water goes through the soil or the media, some improvement of water quality can occur. And so that's where my research is. Constructed wetlands, it's easier to imagine. It's basically a pond with some plants. It can be immersion plants, it can be floating plant or submerged plant. There is also some open water area in which, due to the action of light and photosynthesis processes, algae also naturally develop. Okay, so in this two type of system, in today's talk, I'll focus on benzotriazole. So benzotriazole is this molecule here, so a benzene ring and then that triazole ring here. It's a corrosion inhibitor and it's incorporated into antifreeze liquids in vehicles, for example, and de-icer fluids in airport de-icing pads. And so because of this use, we found it in urban stormwater runoff in the winter, in particular, because this is antifreeze de-icer product, and with salt as a common co-contaminant. But it's also incorporated into dishwashing detergent, and because of this use, you find it quite frequently in domestic wastewater. So it's quite ubiquitously found in the environment. So benzotriazole has a very high solubility and it's quite hydrophilic. In other words, we tend to find it in the water more than in absorbed on fluid surfaces. And it has a pKa at around 8.4 here. There was a nice study done in 2009 by a European group, and they looked at many different trace organic contaminants. That's why you see so many names here on this x-axis in a variety of European rivers. So I like to show this graph just because it was quite a nice overview paper, very large scale that they sampled from. And so what we can see here, what I want to highlight is that benzotriazole is found at a very high detection frequency, which is shown here at about 95%, with an average concentration at around 500 nanogram per liter. So that's quite high average concentration and quite high detection frequency. As for its toxicity, typically the environmental concentration of benzotriazole are lower than the acute and chronic toxic level that are in the low milligram per liter range, but it's a suspect human carcinogen in some countries like the Netherlands. And more recent research has shown that it can affect or create physiological responses in organisms with concentrations that can become relevant, environmentally relevant. For example, it alters the molding frequency in Daphnia magna concentration as low as 2 micrograms per liter. It degrades very slowly, so it's very persistent in the environment. It's poorly removed in wastewater treatment plants, and it has weak adsorption properties. So because of this high use in many different products, frequent detection in the environment, concentration that sometimes are quite high, and it's persistent in the environment, this is a contaminant of concern. Okay, so this is the outline of today's talk. So kind of graphical outline, let me walk you through this. So I've shown, the type of, we've done a few different work on benzotriazole, and I'm just going to show you some of it today. But we work at different space scale, if I may say, from lab work to field scale, and different time scale here, from very short lab work scale, for example, the absorption-desorption study that we did, longer time scale with a photodegradation study that we'll show at the end of this work. And then we work at a variety of time scale, but in the field, in a full-size system, in the tracer test that we did. And so here I'm going to show you work that's relevant to bioretention cell, as shown in green here, and then which is relevant to the context of constructed wetland, which is that aqueous phototransformation study. And I will start with our adsorption-desorption test. So benzotriazole, as I mentioned earlier, it's a winter contaminant. And so in this work, we wanted to investigate the adsorption and desorption potential of benzotriazole in bioretention cell, soil and mulch, like whatever solid surfaces you can find in a bioretention cell. I wanted to look at this as a function of temperature, salinity, and concentration of benzotriazole. So we've worked on a site which is located about an hour north of Toronto, located at the Courtright Center for Conservation. It's a site that started operation in 2013. And so you can see a picture of the bioretention cell here, like kind of close-up picture. You have another one here. This is the inlet. Okay, so runoff from this parking lot is going in the bioretention cell here. And here you have an aerial view. So the system is located basically here. And it receives runoff from this area here, which is in kind of a darker color. You can see that this parking lot here has different colors on the pavement. And so this is because the Toronto Erosion Conservation Authority, which is our partner there for this work, was trying different sustainable practices and different types of pavement and looking at how it can control or reduce stormwater flows. And so here they have concrete, asphalt, parking lots, but also recycled rubber. And so this is what we have here. The drainage area of our bioretention cell is made of recycled tires. And that was very interesting in our microplastic study. And I'm happy to take question of that if you have any, but I'm not going to talk about that today, just for the sake of time. Okay, so we collected soil from this bioretention cell here, but we collected both soil and mulch because many of this system have hardwood mulch distributed on top. And so what I'm going to show you on this graph is the adsorption coefficient. So KF is the Freundlich adsorption coefficient, and OC means that it's normalized to the organic carbon content. And it has weird units. We can talk about this later. It has weird units. But what you should just remember from this graph is that the longer the bar, so the higher this KFOC parameter, then the higher the adsorption capacity of benzotriazole on either the mulch in white or the soil in gray. And we did this at different temperature levels, as you can see here, from 4, 10, 22, and 32 degrees Celsius. What we found here is that the adsorption of benzotriazole on the soil was larger than the adsorption on the mulch. As you can see from the bars from the soil that were larger than for the mulch. We also saw that the adsorption capacity increased as the temperature decreased. And so this is because at low temperature, benzotriazole is less soluble and therefore it adsorbs more. And so that's good news for benzotriazole because we tend to find it in the winter when the temperature is lower. Here are the results for the salinity. And again, I'm showing you this KFOC coefficient. So kind of same graph as we had before for the soil and the mulch with different salinity level. And what we found here is that, again, the adsorption increased slightly with the presence of salt. And this is because when you have more salt, benzotriazole doesn't want to stay in the water, so it's less soluble, so it tends to absorb more. And so again, this is good news for benzotriazole because we tend to find it in urban stormwater runoff with salt as a co-contaminant. And finally, I'm showing you the results for the effect of concentration. And here, just to change the story a little bit, I'm showing you the desorption potential. So H is kind of a hysteresis coefficient, and the larger its value, then the larger the desorption potential. So desorption is when the molecule previously adsorbed on a solid surface, it's kind of released back into the water. Okay, and so we did this at different temperature levels again, and it doesn't matter which of these 4 groups you look at because they all look the same. They have this kind of stairs pattern, and we did it at different concentration level. Okay, and the lowest concentration here is with the black. And what you can see is that as the concentration decreases, you have less desorption. Again, it's good news for benzotriazole because we tend to find it in the environment at lower concentration. Okay, so take-home messages here is that At environmentally relevant concentration, which means lower concentration in urban stormwater, benzotriazole can absorb more under cold climate condition of presence of salt and low temperature. So yes, there is some potential for bioretention cell to retain benzotriazole. However, the actual absolute value of this coefficient was not very high. It was what we would call a moderate absorption coefficient, suggesting that as by itself, this media, which is basically sand with some organic matter, is not the best at retaining benzotriazole, and we could definitely improve this absorption capacity if we added amendments. And some amendments that could work include ferrihydrite, biochars that were shown in the literature to enhance benzotriazole absorption. Okay, so I'm going to stay on the context of bioretention cell, but this time I'm going to present data from a field study. We really wanted to investigate how benzotriazole behaves under more realistic field condition. But as I said, sometimes it's present at very low concentration, and I don't know if at this site particularly we have a chance to see the specific molecule, the data we're ready to look at it and go collect samples. So what we did is that we did a tracer test, or some others call that an injection or a challenge test. It consisted of first injecting water from a cistern, so several cubic meters of water, a little more than 6 cubic meters of water, in order to kind of simulate a runoff event. And then we did a pulse injection of a tracer mixture. So this is the pulse injection, a bucket that you pour at the inlet of the system in a short period of time compared to the total length of the experiment. And in this tracer mixture, we had bromide as a water tracer so that we could check the hydrology and the hydraulic functioning of the system. We injected rhodamine WT, which is a dye that gives this bright pink color. And the reason we use this, there are a few reasons, but one of the reasons is that it helps you visually see when the tracer comes and when it gets diluted. So it helped us adjust our sampling frequency. And finally, we also had benzotriazole and other contaminants that I won't have time to go over today. I'll focus on the benzotriazole story. The actual tracer test lasted about 6 hours. So this is kind of a day of work. And then when we stopped the water at the inlet of this bioretention cell, the water in the effluent quickly stopped as well. But then what we did is that I was very interested in trying to figure out what happens to benzotriazole and this kind of tracer organic contaminants in between rain events, because most of the literature focuses on inlet versus outlet system, determine a % reduction of concentration, but they don't really look at what's going on within the system. And so to do this, we left all our sampling equipment ready to collect effluent samples from the bioretention cell during the next natural rain event. And that happened, about 26 days after our tracer test. So in this experiment, we collected soil samples before and after the tracer test, but I'm not really going to show this today. What I'll show you especially is results from our water samples collected in the effluent of the bioretention cells. Okay, so let's start with the kind of hydrology results using our bromide, which was our water tracer. So here I'm showing you the concentration of bromide as a function of time since the start of the experiment. We had an excellent recovery of bromide, which suggests that we can rely on these results to evaluate the hydrology of the system. In other words, we didn't have major leaks where we would have lost our tracer. From this tracer curve, like this kind of signal in the effluent, we were able to estimate the retention time of the system, and this was 1.8 hours. And so for those who might be in the field of green infrastructure, this is very short. It means the water infiltrated very fast through the system, and much faster, in my opinion, than it should, given that the systems are designed so that they drain the water in 24 hours. Usually the systems are over-designed, and as I mentioned earlier, these bioretention cells, the primary goal is to control the volume of water. Water treatment is kind of a secondary objective that people hope or think is happening. But the primary goal is really to have a fast infiltration of the water so that it doesn't stay and it doesn't create this risk of flooding. But honestly, this is a bit too short, and our local design guidelines document actually in Ontario suggests that a typical runoff event should infiltrate in 24 hours. So clearly, you know, we could keep the water in the system a little bit longer without affecting the hydraulic functioning. We also found that the effective volume ratio was 0.3. So what that means is that only 30% of the bioretention cell soil volume was actually used during the experiment. So we had a lot of dead zones, dead volumes. And so clearly there is an opportunity to use more of this bioretention cell. So not that great in terms of this hydraulic functioning, but at least it's characterized and we can use this information or this context in order to interpret our data for the contaminants. All right, so these were the bromide data, and I'm going to show you a similar graph, but for benzotriazole. Again, this is the concentration versus time since the start of the experiment. We had here a lower recovery of benzotriazole compared to bromide, suggesting that it doesn't behave exactly like water. Something else is happening to it. But yes, of course, I agree, not that much. The recovery was still above 70%. Thanks to our soil sampling data and a specific modeling study that another student, Tim Rogers, did on this bioretention cell, we were able to estimate that approximately 8.8, so 9% of the benzotriazole was likely absorbed to the soil, 16% exfiltrated. So that means that the water went below the bioretention cell towards the groundwater, and then 1% transformed. So as expected from our lab experiment, we did have some but moderate absorption of benzotriazole, and during the tracer test, our model suggests negligible transformation. I would note that this soil absorption potential estimated here is probably an underestimate because at lower concentration, it tends to absorb more. And here we were working at higher concentration than environmentally relevant. Okay, but this transformation potential, I was very interested to try and understand this better. And so the way we approach this is by looking at transformation product. So on this slide, I'm going to show you two graphs, which the first one will represent the concentration on the y-axis in nanomole per liter versus time since the start of the injection. And the time is broken, and we'll have the same x-axis here on the second graph. It's broken because the first part of the graph will be for the actual tracer test, whereas the rest is for concentration that we found in the samples collected during that next natural rain event. Okay, so still bioretention cell effluent samples, but that second part of the graph is for what's going on during the next natural rain event. So here we have a concentration in nanomole per liter, but here in the bottom graph, instead of concentration, I'll show you the peak area for two products, simply because these two products had different isomers that we could not separate. So these are kind of proxies for abundance. All right, so we had many different authentic standards. By many, I mean around 20, I think, for known transformation products of benzotriazole. And this included photo transformation, which is not relevant here because there is no ponding water for photo-induced processes. But the ones I'm going to show you here are the plant transformation products, benzotriazole alanine in purple and benzotriazole acetylalanine, and then three microbial transformation products, the methyl benzotriazole, the hydroxy, and then the methoxy benzotriazoles, which again had different isomers. And so these three were only found in the literature as signature of microbial transformation, whereas these two are signatures of phytotransformation. Okay, so here are the results. Let's first look at microbial transformation. We did find some microbial transformation. If you look at this kind of green curve here, it's hard to see because I've chosen to put the phytotransformation product on the front, but we did have a peak of methylbenzotriazole here during the tracer test and a little bit of hydroxybenzotriazole. And we are confident in these results because we had many field blanks, soil samples, etc. So there was something, but it wasn't that much. The main microbial transformation that we observed was in between this rain event, right? In these 26 days when some of the benzotriazole is stuck in the system, indeed some microbial transformation can happen, forming methylbenzotriazole and then some methoxybenzotriazole. But what was the most exciting was to find that we had significant and fast phytotransformation of benzotriazole through this amino acid conjugation pathway. There is another phytotransformation reaction pathway through glycosylation. We had standards for this, but we didn't find any, suggesting that this, at least at our site, was the dominant pathway. And at the beginning, we were very, suspicious of these results because I wasn't expecting to see such fast reaction, but it's actually consistent with the literature. And there are only 2 papers published so far in this context, which are mesocosms. One is a mesocosm study, one is an hydroponic study. So not the full scale at which we're working here, but they both show that benzotriazole can go through this pathway. And they also showed that benzotriazole acetylalanine, which is the one in pink here, tend to accumulate in plant tissue, whereas the benzotriazole alanine is released by the plant. And that's consistent with what we found, that we found less of the acetylalanine, likely because it is accumulated in the plant tissue. Okay, so take-home messages here. Just to start, this bioretention cell was what I would call a conventionally designed bioretention cell. In other words, it was made of some sandy media, it has plant, but there were no specific amendments to it in order to either support biotransformation of contaminants through co-metabolism, for example, by the addition of biochar, or to store contaminants. Okay, so no amendments for that. And so in spite of this condition, we did found under full-scale condition that there was some absorption, moderate absorption of benzotriazole, some microbial transformation in between rain events, something that no one has really looked at before, and then very fast phytotransformation. But obviously, so for me, there is potential in this kind of system, but we need to work to exploit this potential a little bit better. There are strategies that can be implemented in order to optimize this treatment potential. And first of all, obviously, we need to work on the hydraulics and making sure that the full volume of the bioretention cell is used for treatment. We saw that keeping plants in the system is relevant because they can act either directly or indirectly in contaminant transformation. And we can think about various amendments. First, to better sorb benzotriazole, but second, because organic amendments can release tasty bioavailable carbon for bacteria that can then use it and do co-metabolism for the treatment or the degradation of benzotriazole. Okay, so I'm going to close that bioretention cell story, and I'll stay on benzotriazole, and I'll talk about the kind of that last process that I haven't talked about, which is its phototransformation. So this is more the context of constructed wetland, at least that's how we're thinking about it, what can happen to benzotriazole either in a river, a stream, or a constructed wetlands. But I'm going to show you a lab experiment. And the reason I chose to talk about this today is because it's an opportunity for me to introduce that other aspect of my research, which is on compound-specific isotope analysis and how we can use this technique in order to understand contaminant fate. So CSIA, or compound-specific isotope analysis, relies on the natural distribution and natural abundance of various isotopes. So isotopes are the same elements, but they differ by the number of neutrons, as shown here for hydrogen. And as I said, they are present at natural abundance. For example, of the atoms of carbon around us, 98.9% is carbon-12, which is a stable, light isotope of carbon, and about 1.11% is carbon-13, which is a heavier isotope because it has an extra neutron in the nucleus, but it's still stable. In other words, it doesn't decay radioactively. And then at natural abundance, you also have some carbon-14, but that 10 to the minus 12% compared to all the carbon. So this is, not going to kill us at this stage. So in CSIA, so compound-specific isotope analysis, we rely on the natural abundance of a light, for example, carbon-12, and heavy, stable isotope. And I just want to insist one more time, this is based on natural abundance. In other words, we're not working in any of the data I'll show you when we are not working with certain C labeled or labeled compounds. Okay, so the same way as when we want to know how much stuff is in a sample, we determine a concentration. In CSIA, we determine what we call a delta value or isotopic signature, shown here as an example for carbon. And this corresponds to the ratio between carbon-13 and carbon-12, or a heavy versus a light stable isotope in a given sample for a given molecule. And this ratio is compared to that same ratio in an international standard. So that results out of my lab are comparable to those out of Munich and Leipzig and all the other groups doing this kind of things. It's often a negative number, and it's very small, so it's expressed in per mil, or the unit now is milliurea. And so we're using a very specific mass spectrometer to do this. It's an isotope ratio mass spectrometer that's coupled to a GC, and we have a variety of pre-concentration technique ahead of that. Okay, so how can CSIA help us investigate in situ degradation of contaminants? Or how can it help us investigate reaction mechanism? So typically during degradative processes or transformation mechanism that involve the breaking of chemical bonds, there is what we call an isotope fractionation. And this is because molecules that incorporate exclusively light isotope tend to react faster than those that incorporate a heavy isotope. So imagine that you observe a decrease in concentration over time or over distance. For example, you see that along a groundwater plume, contaminated plume, you see that the concentration decrease. Or over time in a river, you take samples and sample, you see the concentration decrease. If at the same time or at the same place, you see an increase, or I would say more generally a change in the delta value, then this is a sign that degradative processes dominate. Conversely, for non-degradative processes or transfer mechanisms that simply involve the redistribution of contaminants in the environment, for example, through adsorption, volatilization, or dilution, usually there's no significant isotope fractionation. And so if you observe a decrease in concentration, again, it only tells you we have contaminant loss, but you don't know if it's degradative and non-degradative. But if at the same time the delta value remains constant, then that can be a sign that these transfer mechanisms dominate. So this is under ideal condition. But before you can apply this principle to the field, there are several prerequisites. It's absolutely fundamental to do laboratory experiments under controlled condition in order to evaluate if a set of conditions, for example, aerobic biotransformation of the contaminants, actually leads to isotope fractionation. And if this isotope fractionation is significant, reproducible, and we can distinguish it from isotope fractionation of other mechanisms that can happen at the same time. And hence, experiments of this type are done, for example, a microcosm experiment where you collect samples over time, or the phototransformation experiments that I will show you. And so over time, you collect samples that you analyze for concentration, and you can express the concentration as a fraction remaining. So this is C over C0, if you wish. And you also measure the delta value. Then you plot your data like this, and you can model this kind of relationship between the fraction of the compounds, or the concentration, and the delta value through the Rayleigh equation. And as you know the fraction of remaining compound through your concentration analysis, and you know your delta values, you can estimate this parameter epsilon, which is called an enrichment factor. And the larger the magnitude of epsilon, the larger the isotope fractionation. Okay, so we looked at the photolysis of benzotriazole, and we looked at both direct transformation and indirect phototransformation. So in direct phototransformation, the molecule here, benzotriazole, directly absorbs light, it excites it, and then it triggers its transformation. whereas in indirect phototransformation, it's another molecule in the system, for example, nitrate, dissolved oxygen, dissolved organic carbon, that absorbs light, leading this molecule to reach a higher energy state. And this reactive molecule then reacts with your target contaminant benzotriazole, triggering its transformation. So here we looked at the kinetics, this isotope fractionation reaction mechanisms for benzotriazole aqueous phototransformation. And we did this at different pH. Why? Because as I said earlier, benzotriazole has a pKa at around 8.2, 8.6, so kind of around neutral. So at low pH, it's protonated here, but at high pH, this triazole ring bears an anionic negative charge, and that can affect its phototransformation. So let me jump into the results. And so here we're showing the concentration as a function of time since the start of this irradiation experiment for the direct photolysis at pH 5, 7 in red, and 9 in blue. What you can see is that the photolysis was faster at lower pH and even faster in our reaction with hydroxyl radicals. So in the indirect phototransformation experiments, we were specifically looking at the reaction of benzotriazole with hydroxyl radical. And here, these are the pseudo first order transformation rate constants. So basically you put first order model on this, you estimate a K value. And so you can see that this is an order of magnitude higher than for the direct photolysis here. So this is for the kinetics part. And so you can see that, you know, the form of the molecule, whether it's in, or the species, whether it's ionic or not, matters in terms of its reactivity. And so let me show you some of this isotope data. And so here I'm showing you the delta C13, so the stable isotope value for carbon on the y-axis on the left here in black. So this is this one here. And then the delta 2H, so hydrogen isotope analysis in red, and then nitrogen isotope analysis in blue. For one experiment, the one done at pH 7. And so this is all versus time since the start of the experiment. What you can see here is that we had negligible isotope fractionation on hydrogen. It was moderate on carbon. It was very high for nitrogen. And as I said, you can express this extent of fractionation using this epsilon value. So the higher the epsilon value, as you can see here, then the higher the isotope fractionation as observed here. And so we did this for pH 7, 5, 9, and also for the UVH2O2 experiment. UVH2O2, this is our way of generating hydroxyl radicals. So what we observed is that benzotriazole aqueous phototransformation during direct photolysis led to some carbon isotope fractionation that increased with increasing pH. We only found relevant hydrogen isotope fractionation at pH 5, and it was very significant when benzotriazole reacted with hydroxyl radical. And then we found variable nitrogen isotope fractionation with the direct photolysis experiment, and none in the reaction with hydroxyl radicals. All right, so this looks a bit full, but I've added here what we think are our main reaction pathway. And so we use the kinetics data, we use the carbon, hydrogen, and nitrogen isotope data, and some transformation product analysis to understand what the reaction pathway could be. And so I've shown here benzotriazole in the protonated form, and this is its ionic form that's only relevant at pH 9. And you see different colors are for different pH value here. So first, you know, like globally, what we found is that when benzotriazole reacts with hydroxyl radical, we found some hydroxybenzotriazole and some dehydroxybenzotriazole as our main transformation product. And these, along with very significant hydrogen isotope fractionation and moderate on or very low on carbon and none of nitrogen, suggest that we had an hydroxyl radical addition to the benzene ring, which is triggered by first a CO bond formation, and which is kind of the slow step, and then a fast carbon hydrogen bond cleavage. And this is consistent with the fact that, for example, no nitrogen is involved in this reaction, which is why we see no nitrogen isotope fractionation. And then under direct photolysis, we observed that we had a nitrogen-nitrogen bond cleavage followed by a carbon-nitrogen bond cleavage here, and that this was led to an intermediate that was stabilized via two routes, forming aniline on one hand and triaminophenol, and then the dimerization process here. And this is consistent with the nitrogen, more significant nitrogen isofractionation that we found. Okay, so just to sum up, we did find a pH dependency of benzotriazole photolysis. We showed here that multi-element CSIA, so stable isotope analysis of not just carbon, but carbon and hydrogen and nitrogen, can help us decipher reaction mechanisms. And it can help us now identify the aqueous phototransformation of benzotriazole in the field. And so that can happen, for example, in rivers. We also found that the benzotriazole was photodegraded faster at lower pH. And so that suggests that, for example, constructed wetlands, if I come back to, how we kind of use this information for the green infrastructure, the remediation side, it suggests that wetlands would probably not be the best for this kind of molecule, because wetlands tend to have algae developing in them that oxygenate the water during the day, but also they naturally increase the pH. Whereas here you would want a lower pH in order to enhance the fast phototransformation of benzotriazole. But we've done quite a lot of recent work with duckweed, looking at the uptake of benzotriazole by plant and its transformation. So, there are other ways to use wetland for benzotriazole transformation. So I hope I showed that CSIA was a powerful tool to conclusively identify transformation and also help decipher reaction mechanism. And CSIA is a tool that's applied traditionally for groundwater contamination, for for legacy contaminants. And so our group is one of the few where we're applying it to new molecules like benzotriazole and new environments like wetlands and this surface water environment. Okay, I'll just conclude by saying that there is potential in this green infrastructure for water treatment, but this potential is not fully exploited yet. And I strongly believe that by better understanding the hydrological processes and biogeochemical processes that control these different transformation reaction mechanisms, this new knowledge can really help us inform which green infrastructure can work, how to design them, and how to operate them so that we can get more efficient and more robust water treatment. Before I conclude, I'd like to mention that this work, of course, is done by many students, graduate students, and postdoc, and those that contributed to what I showed today are listed here. And we have, of course, many academic and private partners to fund this research. And now I'm going to stop, and I'll be very happy to take any questions.
Oh, thank you very much for a very interesting talk. Lots of things to think about. So I'll open the floor for questions. Unmute yourself if you're going to ask a question.
I'll ask a question. The stable isotope work for tracing all of the things that you do, the degradation, the biotransformations, et cetera, that's potentially very, very expensive in a true reactor. Do you ever think about scaling up to that kind of size? In other words, a true constructed wetland or whatever else, or do you have to maintain it all in a very carefully controlled environment?
No, it's very easy to do. I mean, very easy. It's completely possible to do this in the field. And so it's used a lot. Maybe I can give just quickly a historical context here. CSIA is used a lot in the tracking of legacy contaminant, as I said, like benzene, toluene, chlorinated solvent, and it's used in the field. So what we do, I mean, you have to do this lab experiment so that you understand first if you can expect fractionation or not.
Right.
If you don't do this, then there's no point in going to the field because you can't interpret the field data. But going to the field means collecting samples from the field and analyzing them for CSIA. And we've done this actually for wetlands, for pilot wetlands at a contaminated, highly contaminated site in Brazil. And our partners there, so this is Corteva, a big chemical company that is trying to close the site. So they collected samples in this wetland, they ship it to us. And what we do is that we separate different chemical groups in the lab by solid phase extraction. We pre-concentrate the samples and then we analyze them. And so we've done it. And in this work, we showed that there is, in that case, 2,3-dichloroaniline biotransformation. And this is very important for partners at the site because they must provide various lines of evidence, with conclusive evidence that transformation is occurring. Not necessarily biotransformation, but any bond breaking to less harmful products, and not just contaminant loss. So it's one of these advanced tools with molecular tools that and others and modeling that can be used, but this is the tool that our group contributes.
Right, and I really wasn't referring to the fractionation. I understand how that occurs. What I'm saying is, if you're going to purchase a C13 labeled compound and then introduce it into wetland, how do you get any sort of measurable concentration back out after any sort of period of time?
Okay, so again, we do not introduce any labeled compounds.
Right. So that was my primary question.
I'm sorry if I misunderstood it. Yes, indeed. So this is really working at natural abundance. We're not labeling anything. It's really due to these natural processes that molecules that exclusively have light isotope, they can run fast, they can react faster than the molecule that contains a heavy isotope and that goes a little bit slower into reaction. So that's why we see this kind of enrichment, we call that an enrichment in the heavy isotope in the remaining not yet degraded pool of contaminants. So it's all natural abundance.
Right.
There's no issue with that.
I asked because I do know people who've created model systems, meaning smaller versions of wetlands and such, and introduce the stable labels that way and trace things. But I could not see you using that in a true field environment in terms of just expense alone. Forget the fact that you are quote unquote introducing a contaminant, but just the expense. So, okay.
No, you're completely right. And we do similar work in our lab. So we don't use 13C labeled compounds because I wouldn't want to introduce this in my lab. It's forbidden in my lab because I wouldn't want to contaminate, my mass spec, which is working at natural abundance. But I do have also all the instruments to work with radio labeled compounds, which is kind of old school now. Now, as you were saying, there is more 13C labeled compounds, and with this fancy high resolution mass spec, we can see so many different things using this kind of labeling. But we do work with radio labeled compounds, and we do the kind of experiments you described. And obviously here, we're never going to inject a radio labeled molecule
into the environment. Yes.
Yeah.
So I have a more general question. The approach that you use when using wetlands has been, always thought to be a good thing. But when you get biotransformation or phototransformation, there's always the possibility that you create new toxins that are worse than the original. And so for the compound you looked at, that may not have been the case, but in some cases that will be. And so the approach that you're using where you look at the effects of pH or salinity and so on, do you think that can be used on a larger scale? Say you are creating a more toxic compound, can you then modulate how that's metabolized in your artificial system or the wetlands that you're creating to actually move away from producing a more toxic compound metabolite?
Yeah, thank you very much for this question. This is a great observation, and you're absolutely right. I think people are aware that some of this transformation product can be more toxic than the parent molecule. And a good example is triclosan. So triclosan can also phototransforms. And one of its, or several, I would say, of its phototransformation products are more toxic than triclosan itself. For example, it can form dioxins, 2,7 and 2,8 DCDD, so very toxic dioxin, and it can also form 2,4-dichlorophenol, which is on the US EPA list of priority contaminants. So in some of our work, one of my first PhD student actually, who graduated now, we looked at, so we did kind of similar lab experiments without the CSIA part, but our objective was to simultaneously find conditions that would lead to the highest rate of triclosan phototransformation while minimizing the accumulation of toxic byproduct. And so to address this, we used a design of experiment approach, so an experimental design that had, that used a second order models, kind of a surface response design. And so we did many different experiments, very organized so that we could have enough and consistent data to do our modeling. And so we find condition of nitrate concentration, dissolved organic carbon concentration, and pH that would lead to this dual objective of accelerating or like getting, the fastest transformation of triclosan while minimizing the production of this product. And so we did find this, and that was interesting because we found that nitrate concentration didn't really matter. And we thought it would because nitrate can create some radicals that can react indirectly with triclosan, but we needed low dissolved organic carbon concentration, mainly because it acts as a light screener. If the water is turbid, you have less light penetrating. And we also found that pH, the higher the pH, the fastest the phototransformation of triclosan, while the least accumulation of toxic byproduct. And so that helped us think, okay, in the field now, if you try to translate this to the field, it means that you would need some sort of a open water wetland where algae, for example, can develop, not too much because then you have light screening, but in a way that it can increase the pH of the water. Or you can think of some other system where you pass the water first through some, carbonate media to increase the pH. I'm a strong believer of treatment unit concept. I come from a chemical engineering background. I teach this morning before I came. I was teaching, biological wastewater treatment. So in the wastewater treatment, you have different units and each is designated to a specific removal objective. So I believe in this in the field because a big pond with plants is not going to do the job at removing all the contaminants because they all need different conditions.
And of course, the obvious question that always comes up when you use any kind of bioremediation, you're concentrating the chemicals now in what is arguably a contained system, but what do you do with it in the end when it's completely saturated?
And it's funny because we were talking about this at the conference. We had a project meeting because the partners were there, and one of them was worried about the accumulation of contaminants in the plant material. Because when you work at super contaminated site, you have both some passive uptake by plants and some active uptake. So you can have contaminants accumulated in plants. So I think this question is relevant for very, very contaminated site where it really must be evaluated carefully. However, when you look at trace organic contaminants, I also believe that a system like a bioretention cell, for example, okay, taking this as an example, will fail because it will clog before it's full of organic contaminants. Yeah.
Any other questions?
Hi, Helmut. Can I ask you a question?
Sure, go ahead.
Hi, Elodie, this is Phil Democritou. Thank you for the wonderful presentation. I was wondering if your retention cell can be an effective way of removing micro nanoplastics from the environment. Myself and others here at the EOHSI, we have interest in micro nanoplastics, and of course, the main focus of our research is understanding the human health implications. But and also, we're thinking of a Superfund application next year, and for the NIH Superfund program, and we need to include engineering projects, how we can solve the problem, not just to start the potential effects on humans. So I was wondering if your retention cell can be micro nanoplastic specific, and if you cannot that specificity can be an effective way of reducing the concentrations of microplastics in water media.
Yes. So let me answer your question
Well, here it is.
here.
Okay.
Oops.
I cannot see that.
Yeah, 1 minute. Let me slideshow here. Okay, do you see my screen now?
Yes, yes.
Okay, so the answer is yes for microplastics. So we've worked on bioretention. So this bioretention cell, actually, we've studied it quite widely, and we found, which is found in the literature, that it works very well for total suspended solid removal. Obviously, it's a physical filtration problem, so that's why it works very well. You know, the particles are just stored in the system. So that led us to hypothesize that microplastics could be also removed in this kind of system. And so, indeed, as I show here, one of our first paper by Sylvis Brackman showed that there is significant reduction in concentration of total suspended solid between the inlet and outlet of this bioretention cell. Okay, so we wanted to look at microplastics. And I want to insist that microplastics are defined as plastic particles between 1 and 5,000 micrometer. Below this is the nanoplastic. What we did is that we look at the 100 micrometer to 5,000 micrometer size fraction. Why? Because it's easy to analyze. By easy, I mean that we need an army of undergraduate students to count the particles one by one under a microscope. But we give them other projects, don't worry, they are happy in our lab, but it is a tedious process. And below 100 micron, we need to use some other techniques. So anyway, so we did this, and just to go fast to answer your question, yes, we found, so this paper by Kelsey Smith et al., who is one of my PhD students, was the first exhaustive study. There was just one study with 3 samples before, but this is the first study that's now cited quite a lot, showing that bioretention cells are very efficient to decrease microplastic concentration in this size range. And so I want to insist, so, we look at other things, not just the amount removed, but also what they contain, the polymer type, the shape, etc. But I'm not going to say that. Then we started at 100 micron. Then we did 20 to 100 micron. And this is in prep. And we did this by micro FTIR mapping. But below 20 micron, I don't know how to do it yet.
Right. Yeah, but the big issue, at least from the emerging literature, is the nano size fraction of microplastics, and those are the ones that will end up in the water we drink and the food we eat. And the size specificity is very important. I mean, I share your concerns in terms of method development. There are some emerging methods, for instance, coupling pyrolysis with GCMS, it seems very, very promising. And there are some other groups. Yeah. Oh,
I'm sorry, I didn't mean to interrupt.
Yeah, there are some emerging techniques that they can be used to quantify down to the nanoscale. So we have some projects, and we're thinking of developing these approaches even in-house or through collaborators. But I would love to include you as part of this Superfund team that we're formulating as we speak for a potential NIH Superfund, and because we need to come up with approaches to remove microplastics for specific environmental media. So if you have any interest, let me know.
Absolutely. It took a long time to develop the method and validate them. And I'm careful really in my work. I mean, I do stabilized top analysis. I have an analytical chemistry background. It takes a long time to get to a good analytical technique that's reliable. And so now I definitely want to exploit all the work we've done on the microplastic, and now with the micro FTIR, so we've done quite a bit on this, and we're doing the mapping in the small size range. And I strongly believe in what you mentioned, pyrolysis GCMS for the nano size, but also because in our work with whether we use micro Raman or micro FTIR, we cannot see the rubber particles. We cannot see the black particles. And that's a huge problem for the type of media that so far I've been working on, because we find so much what we think is tire wear, tire particles, but we can't prove it.
Right. Yeah. No, it's, and also I can bring you in contact. I was in McGill University in September. There is also an emerging group there looking on plastics in agriculture.
Yes, we do this too, actually, with friends.
Yeah, and we we're writing a paper out of that 2-day symposium that was there. So yeah, it's a lot of work on plastics and microplastics in general. So as I said, if you have any interest, we will invite you and see how we can. I know NIH allows partnerships with Canadian universities, so that can be really.
Yeah, I would love that. Thank you.
Okay, thank you.
Well, that's an exciting possible outcome of the seminar. And are there any other questions? Sorry, any other questions? Okay, if not, we'll thank Dr. Passeport again for a very interesting talk, and hopefully look forward to seeing you here in New Jersey at some point.
Yeah, hopefully. I was let in the US this week, so I was quite happy.
That's
my
good.
US green card application, they say,
Okay.
they didn't even know.
Well,
Thank you very much.
Yeah, thank you so much for the invitation, Nancy, and Helmut for all the introduction. I really, I had a great time, great discussion. Thank you very much.
Thank you.
Thank